Welcome to Eye to Market, a web series designed to help you explore and navigate the successful path to market for your ophthalmic innovation. Welcome to Eye to Market. My name is Brian and I'll be your host today. In this episode, we're going to talk about formulation. How do we create a formula that's stable and workable for your clinical trials? Let's dive in.
So when you start thinking about creating a formula, what are some of the things that you look at? Are there key factors about different excipients or processes of development that help you achieve more efficiently a stable, safe, and effective product versus let's just throw a bunch of stuff in the pot and see if it works?
Well, it has to be done logically because not all excipients are going to play with each other, right? And so you have to think of things like, is it an ionic compound, right? Are you going to, if you're going to say in my world with contact lenses and dry eye, I'm looking at a lot of surfactants, right? So are you going to use an ionic surfactant? And if you are, it's going to be a cation, is it going to be an anion? Do you need a non-ionic surfactant, right? Because each of these is going to play a little bit different. When you add different excipients to it, for example, if you decide you're going to buffer with phosphate, yet you put calcium in it, all of a sudden your calcium flocks out of your system. These are known things, right, that are out there. So a lot of it comes with doing the research and not being surprised by, so the different things matter. So how am I going to buffer a system? Am I going to put borate in it? Am I going to buffer it with citrate? Am I going to put phosphate in it? Every one of those is going to make a difference to the ocular surface itself, right? And you have to make sure that it's going to be stable on the surface, that you're not going to end up with a toxicity relationship on the surface. You have to make sure that it's going to play with the other things, that you're not going to flock out your other excipients that you want in the product as well. So there's every single molecule has to be considered. So if you need a surfactant, you have to put all the surfactants on the table and then say, well, this number is used in eye care. Do I want to use the ones that are used in eye care? Right? Mm-hmm. Am I going to, in my case, I went outside of eye care to start using a different type of surfactant to get a different result. It's not a fast process. I've been working on one of the molecules I'm working on. It's taken me 3 years to get to the point where I'm at right now with it. How are you going to be able to filter it? That was one of the pain points that I talked to you about. The molecule that we're currently talking about right now is I can't filter it. So I had to think about what's the smallest filter I can use to take clumps out, right? Yeah. But can we autoclave it? How else do I get it there, right? But that's going to affect production, and that's why I pulled you in. How are we going to do this? Can we scale it?
Right? Yeah. So how do you think through the product specifications, the analytical methods? What are some of the things that come to mind as you start thinking about developing these protocols, test methods, specifications to ensure that what you're delivering to a patient is what you initially envisioned from a solution perspective?
Well, you have to make sure that the product you end up with is the product you thought you ended up with. And so you have to make sure when you get done that you actually ended up with what you intended to get. And so that's where validation comes in. You have to validate in the lab, running bench tests, whether it's HPLC or whatever testing you need to do to say this is what's in it. But then it has to be an ongoing process, right? So it has to be something, again, working with your manufacturer. Can they validate it too? Because we're going to need to. It's not like it's one and done and it's like a checklist and that's off your list. You're having a product that goes on for years. You have to make sure you keep continuing to make cookies instead of cake, as you go down the road with this. And so you better start pretty early with what the validation tests are. And the products that I'm bringing to market right now are fairly simple in comparison to other products. I always think like whatever you throw in there, decide how much you can eliminate out of there. And so the more simple you can make it, the better and the easier it is going to be to validate the product or the process.
Tox studies, animal studies, how do the performance of them, the results of them, how do they inform your decisions? How does the feedback from those studies impact your process and what that evolution of the product will be, whether it moves forward or you go back to formulation? How do you start looking at those and interpreting?
So first of all, let's back that up for 1 second, because there's a
Hmm.
question before that I think that needs to be answered. First, whatever product somebody's working on, they're going to have to say, do I need those studies? And okay, so reasonably, if you're going to put it on a human, you're going to need those studies. But then the question comes, are they animal studies or are they cell line studies? How are we going to do this? Because there is a segment of society that really doesn't want animal studies, right? And so, what's your target market, right? Are you going to want to be able to make the claim that you didn't do animal studies on this product, right? And if you're
Hmm.
not going to do animal studies, what are you going to do them on, and how are you going to? And I'm not going to say I'm an expert in that field, but it is definitely many conversations that I've had, because there are ways around that. I've never done that. But when the tox studies come back, first of all, you better know when you send out your tox studies, you better have done enough internal data to know what your tox studies are going to be when they come back.
At what point do you typically look to apply for your patent? Is when you've got your final formulation and you're ready to move on from tox,
Mm-mm.
or are you when you've got a concept? I mean, when does that patent application start?
Well, as soon as you get that idea, that better be your 2 a.m. Google search. Here's one thing I absolutely know: the minute I have an idea, it is not new anymore. So it's always a matter of either you have to be an expert, or become a content expert, or know a content expert. Those are your three choices, right? And so you got to start looking through the Google patents. What's out there? Know your landscape. What is out there? Is my idea actually patentable? Because there's a lot of times where there's not a product on the market, but the patent is actually there. And then you have to think, what do I need to do to make this a patentable idea? So, for example, I'm working on something now, big idea. It is not patentable as a formulation. It's patentable as an a patent use patent, right? So
Okay.
it's a new use for something that's already out there. And then you have to think about who has the bigger pocket. So if your patent is owned by one of the giant, not owned, but like the original patent's owned by a giant, are you going to be able to patent it? Who has bigger lawyers? Are they going to look at, there's a lot of landscape that goes in there. But you better start thinking about, is my idea new, novel? If it's not, that does not mean that you can't go to market, right? Like,
Hmm.
I have something, Brian, we've been talking about newish thing. We haven't talked about it too much yet, but it's really not patentable. It's really not a new idea, but it's definitely something that the market needs, right?
Hmm.
I got the funding. Let's put it in a bottle. Let's sell it. And then we're just about what it is, right? So there's also something called a trade secret. So is it something that you're going to patent, or is it a trade secret?
The key point, I would say, a good formulation is really about finding the right balance between efficacy, of course, safety, and stability, while also ensuring patient comfort to the eye. So this is really the good formulation for me. And if I go a little bit more deeper on that question, in ophthalmology specifically, this means that the API, the drug substance, remains stable in a challenging environment because it's mainly composed by aqueous, often oxygen sensitive. We need to be sterile, so without preservatives, because we know that it's toxic on the surface of the eye. So that's really the main point for a good formulation.
Thank you. The manufacturing is one piece of it, right? And how do we take it from this small batch and make it bigger? And what's that manufacturing process going to look like? And can we do it commercially versus or pilot size versus the lab? And what's the differences there?
Yes.
But
You're right.
another piece that you talked about was initially was being able to create a formula that was stable and held up over time. And the way that we do that, the way that we understand if we've done that, is through analytical methods.
Mhm. Yes.
So talk to me a little bit about that validation or that development process that
Mhm.
takes place early in the sequence. And what are some of those stages through the product development life cycle, right? At
Mhm.
what point do you need a verified method versus a validated method or a tested method? What's that evolution look like?
Yes, you're right. We need very strong analytical services to support all the formulation works and pilot batches. So the fact is, we need methods that are not accurate, only accurate, but also stability indicating, and of course, ready to be transferred to the industrial site. We need to analyze the eye drops because we are talking about eye drops at every stage of the development and set the right release specification to ensure compliance. So yes, analytical part is really challenging because you know that better than me also, Ryan. We are working with very sensitive APIs, very low concentration, and increasingly complex formulation. I mean, viscosity agents, emulsion, suspension. So all of this makes the analytical work more and more demanding. And we need to investigate in strong analytical methods. It's really important.
We've talked a little bit about what needs to be done. When does this need to be done? When's the right time for one of our innovators to reach out to a CDMO and request support from a facility like yours?
Yeah, I will definitely recommend those innovators to reach out to a CDMO as soon as they have a conclusive proof of concept results or conclusive preclinical results, really, before moving to the first in human batches. As an example, I can mention that here at Unither, when we work on a proof of concept formulation of our clinical stage, we always think in parallel about the industrial manufacturing process. It helps to anticipate any constraints and avoid redesigning the process. So definitely getting a CDMO involved early would be the ideal. We'll be able to guide them also on process design, on sterilization strategy, and even the regulatory aspect. Uh, we know that we can lose time with that. And so, yeah, bring in CDMO the earliest possible. So for sure, for sterile manufacturing products, we must collect many information regarding the formulation. Is it filterable? Is it highly viscous? Can it be heat sterilized if it's a gel? What are the storage conditions? Um, what else? We have many questions to ask. Which market, of course?
But then we have the client who comes to us and says, hey, I've got an API, but I got nothing else. How does the information we're requesting from that client differ from the other client? What are some of the key things you're looking for when you're looking to create a formulation for a client?
The first question will be for which disease it is. Because
Mm-hmm.
if it's for the back of the eye, if it's for the front of the eye, we won't use the same excipients. We will help the molecule, small, bigger biological molecule. We will need to discuss a little bit more deeply regarding which disease, which API is it, toxicity of the API. If it's just a matter of optimization, we will just try to understand what kind of optimization we would like increase the residence time over the eye. We can add some viscosifier agent. Um, it's always depending on, yes, on which therapeutic area is working on.